Hello dear students, welcome to BioDogma. Hope you are doing well. We know that multicellular organisms are evolved from unicellular organism. But do you ever think why? Let's understand the story of metazoa evolution. Origin of metazoa is very interesting and it creates a new vista in biological world. In unicellular organism, the single cell performs all the biological activities through these cell organelles, and the unicellular organism is all in one in this type of structural organization. Okay, but in multicellular organism, the life originates from a single zygote and it divides by rapid mitotic division, and the stem cell are differentiated into different types of cells. Okay, each and every types of cells has its specific function. Muscle cell for movement and the nerve cell for communication procedure. And here the single cell divides and form a cellular mass, and a group of cells perform the similar function to form the tissue and then the organ structure. This organ actually builds up to form the organ system structure. Okay, these carry out the life process in the multicellular organism. The unicellular organism has very limited surface area to volume ratio. So these are unable to expand beyond their limit. Okay, you can understand আমরা এই ইউনিসেলুলার অর্গানিজমকে ইচ্ছা করলে ইচ্ছে মতো বাড়াতে পারবো না, এর একটা নির্দিষ্ট লিমিট আছে, এটা এর একটা ড্রব্যাক। ওকে, কিন্তু আমরা যদি if we see here the multicellular organism, in multicellular organism the cells are divided and its journey starts from the single zygote. Okay, as it proceeds the cleavage, the volume of that cell gradually decrease. So the cell volume is very small in case of multicellular organisms. Okay, they divide rapidly in their body, and as a result this type of organism can increase its size, they can grow. Okay, it is actually the very better advantage for the multicellular organisms. Okay, here the single cell is everything, okay, for unicellular organism. It has huge amount of workload. It has to look after and perform all the biological process. So if injury occurs in this organism, what happens? Then this type of unicellular organism died. Okay, it cannot be, there is no option for its replacement. So unicellular organism has very limited lifespan. But if we consider the multicellular organism, if each and every type of cells has specific function, that means the division of labor. Okay, all the biological function, okay, or cellular function are equally uniformly distributed among various type of cell. So this cell has not huge amount of workload. They are very flexible. Okay. As a result, okay, this type of cell, in this type of cell, if injury occurs and a few cell dies, okay, this type of cell can be replaced by cell division. Okay, this type of single cell is always in stress. That means certain environmental change like pH, temperature may hampers its biological process. But multicellular organism has different type of system to overcome this situation. That means environmental changes. Okay. So the problem of unicellularity is solved by multicellular concept. কিন্তু এখানেও এক প্রবলেম, মাল্টিসেলুলার অর্গানিজমে যে ডিফারেন্ট টাইপস অফ অর্গান ফরমেশন হয়েছে, ওকে, কেন তৈরি হয়েছে? হ্যাঁ, টু ইনক্রিজ দ্য অর্গানাইজেশনাল কমপ্লেক্সিটি অফ দ্যাট অর্গানিজম, because the multicellular organisms are too much advanced. Okay, different types of system performs various function, say as for the circulatory system. Okay, the heart performs the transfer of nutrients and different molecules into different parts of the organism body. Okay, for this purpose. ওকে, হ্যাঁ, এখন অর্গানিজমকে কি করতে হবে? এই যে ভেরিয়াস টাইপের অর্গান ফরমেশন হয়েছে তাকে তার বডির মধ্যে প্লেস করে দিতে হবে, জায়গা করে দিতে হবে। এটা একটা মাল্টিসেলুলার অর্গানিজমের ক্ষেত্রে সবচেয়ে বড় চ্যালেঞ্জ। ওকে, ধরো তোমার ফিজিক্স, কেমিস্ট্রি, বায়োলজি, ম্যাথামেটিক্স, ওকে, এই সমস্ত বই যদি তুমি হ্যাপাজার্ডলি রেখে দাও, যখন তোমার প্রয়োজন হবে তুমি কি একটা সিনক্রোনাইজড ওয়েতে এই সমস্ত বুকসকে তুমি খুঁজে পাবে? তার জন্য তোমাকে কি করতে হবে? সেই সমস্ত বইগুলোকে, হ্যাঁ, তোমার বুক সেলফের মধ্যে হোক বা পার্টিকুলার প্লেসে একটা ওয়েল অর্গানাইজড ম্যানারে গুছিয়ে রাখতে হবে, যাতে তুমি তোমার প্রয়োজনে সেটাকে সিনক্রোনাইজলি ইউজ করতে পারো। তাহলে এখন অর্গানিজমের সবচেয়ে বড় চ্যালেঞ্জ কি? এই ডিফারেন্ট টাইপসের অর্গানকে বডির মধ্যে প্লেস করে দিতে হবে। তাহলে প্লেস তো এমনি হবে না। যদি একটা মাল্টিসেলুলার অর্গানিজম এ গ্রুপ অফ সেল মাস হয়, একটা সলিড বডি স্ট্রাকচার হয়, ওর ফ্লেক্সিবিলিটি, হ্যাঁ, কখনোই ফরমেশন হবে না। অ্যান্ড দ্যাট ক্যান নট অ্যালাউ the position of the different types of organ in the organism's body. Okay, how the multicellular organisms solve this type of problem? This is the major challenge for multicellular organism. For better understanding, my dear student, today I discuss about the different germ layers and coelom. Fertilization leads to formation of zygote, and zygote undergone the rapid mitotic cell division, and by cleavage it forms the solid cell mass, that means morula stage, and after that it forms a hollow ball of cells, that is blastula. Okay, and these cells are the blastomeres, and in this hollow cell mass a central cavity is formed, that is the blastocell cavity. Here you can see this is the blastocele cavity. Okay, and after that the blastomere shows the gastrulation procedure. Okay, and at the point of where the sperm enter, just opposite to it a particular pore is formed that is known as blastopore. Is it clear? And the cells are invaginated through this blastopore, and these are enfolded, form a different types of layers: outer ectoderm, inner endoderm, and a specific cavity inside the endoderm that is archenteron. Okay, গ্যাস্ট্রুলেশন প্রসিডিওর এবং ব্লাস্টুলা ফর্মেশন তোমাদের কাছে খুব ক্লিয়ার হয়ে যাবে। এটা একটা ফার্টিলাইজড এগ, it undergoes the cleavage procedure. Okay, this is the first cleavage pen and followed by the division, and it forms a cell mass that is the blastula stage by rapid cell division. And this blastula forms a blastopore cavity. These are the blastomere cells. You can clearly see this. Okay, and it performs a specific morphogenetic movement. Here you can see the blastopore position. This is the position of the blastopore. Okay, is it clear? After gastrulation, the cells are invaginated and forms the archenteron structure, and this archenteron is actually the primitive gut, that means the digestive tract or alimentary canal of that organisms. Okay, you can clearly see that different types of early embryonic layers or germ layers are produced. Okay, this is the ectoderm. Okay, this is the mesoderm, and another type of germ layer, this is the endoderm, and this hollow cavity is the blastocele cavity. The organism in which only two germ layers are produced, the ectoderm and the endoderm. Okay, this is the endoderm portion and this is the ectoderm portion, and in between this ectoderm and endoderm an undifferentiated layer, that is mesoglia, is present. This type of organisms are known as diploblastic organisms. Okay, generally the coelenterates, that means Cnidaria and Pomgeli, Ctenophora, are the diploblastic organisms, and where in between ectoderm and endoderm third layer, that is mesoderm, present, this type of arrangement is known as triploblastic. From platyhelminthes to chordates, all organisms are generally triploblastic arrangement. In case of diploblast animal, in between outer ectoderm and inner endoderm, the undifferentiated non-cellular, okay, mesoglia is present. Actually, okay, it is generally composed of the water molecules, okay, and the fibrous protein is present, okay, as collagen, okay, and also heparan sulfate proteoglycan are present in between these ectoderm and endoderm. Okay, these are the major components of the mesoglia layer, and this mesoglia is generally acellular in nature, but it contains some amount of muscle fiber and nerve fiber. But these are not actually the cell. So this is acellular in nature in case of diploblastic animal. Okay. And this type of mesoglia acts as a hydrostatic skeleton. Okay. This hydrostatic skeleton helps to maintain the structure of the organism, okay, and it maintains the shape, structure, and gives the flexibility of that organism. But in case of porifera, okay, the outer dermal layer, okay, this is the pinacoderm layer. This is the pinacoderm layer, and inner gastral layer, this is the coanoderm layer. Actually, this is made up of coanocyte cell. In between this outer dermal and inner coanoderm layer, third non-cellular portion is present, that is the mesohyl layer. Okay, but this mesohyl layer is quite different from this mesoglia part. Okay, this mesohyl is actually gelatinous and non-cellular matrix. Okay, this is composed of collagen fiber and fibronectin like molecule. Okay, and here the major part is the galactin. Okay, this is the major constituent of this mesohyl portion, and another molecule present, that is dermatopontine. Okay, this is present in very less amount. This mesohyl or the non-cellular gelatinous matrix helps in cellular adhesion, signal transduction, and cell growth. Okay, actually sponge do not have true tissue structure. This is multicellular non-tissue grade organization. তোমরা যদি স্পঞ্জের বডিকে স্মাশ করে দাও এবং ওই সেলকে ডিফারেন্সিয়েট করে আলাদা করে আবার সি ওয়াটারে রেখে দাও, তখন ওই সেলগুলো আবার এগ্রিগেট হওয়ার চেষ্টা করবে। ওকে, actually these do not forms any organized tissue structure. In between pinacoderm and coanoderm, the mesohyl is present, and in mesohyl different amoebocyte cells are present, but these cells are loosely organized. Okay, the sponge, that means porifera, do not show the actual or true gastrulation procedure. So in sponge, true ectoderm and true endoderm is not. That means the germ layers are not formed, okay, which is the key features of diploblastic organism. So the sponge, that means porifera, is not considered as diploblastic organisms. And in case of triploblastic organisms, the mesoderm is present in between ectoderm and endoderm. Okay? And the from platyhelminths to caudate, all are diploblastic organisms. And here these germ layers, that means ectoderm, mesoderm and endoderm, forms different types of organ system. Say as for the outer layer forms the epidermis of skin and neuron, nerve cells, okay, and the central nervous system portion. Endoderm layer, inner layer, forms the stomach cell, lung cell, that means alveolar cell, thyroid cell, etc. And from mesoderm, okay, it forms the notochord, it forms the different types of blood cell, connective tissue, bones, cartilage, okay. And from this mesoderm, the coelom is produced. So the question arises, what is coelom? Actually, coelom is the space between the body wall, this is the body wall, okay, this is the body wall portion, and the alimentary canal, okay, that is the digestive system or gut, and this portion is the gut wall, and this is the body wall part, okay. It lies in between the body wall and the digestive system part. Coelom is actually secondarily modified mesodermal body cavity. The coelomic cavity lining must be formed by the mesodermal cell. Okay? Coelom has to fulfill the condition of perivisceral cavity. From this mesodermal cell, the inner visceral peritoneum, okay, is formed. This is the visceral peritoneum layer, and the outer parietal peritoneum is formed. Okay, both these visceral and parietal peritoneum is one type of serous epithelium. Okay, this serous epithelial structure is derived from these mesodermal cells. Okay, the outer parietal peritoneum is actually the abdominal cavity, and the inner visceral peritoneum, this is the inner visceral peritoneum, okay, it is the internal portion of the, just it present in internal position just outside the gut wall. The visceral peritoneum covers the internal visceral organs, okay, and this cavity, that means coelomic cavity, is filled with a specific type of fluid. This is known as coelomic fluid. It is a fluid filled space, and all these internal organs, okay, like heart, okay, like heart, lungs, all are remain suspended, and these types of organs are separated from body wall. ওকে, ধরো তুমি জাম্প করছো বা তোমার বডিতে সাডেনলি একটা বাইট থেকে ধাক্কা লাগলো, তখন আমার ভেতরের যে ইন্টারনাল ডিফারেন্ট টাইপস অফ ভিসারাল অর্গান আছে, কিডনি, লিভার, লাংস, প্যানক্রিয়াস, এগুলো কখনোই ড্যামেজড হয় না। ওকে, এই যে এক্সটার্নাল ফোর্স এর উপরে কাজ করলো, দিস টাইপ অফ এক্সটার্নাল ফোর্স can be overcome, okay, by the coelomic fluid. This coelomic fluid actually works as a shock absorber, just like that. এই shock absorber কি হয়, ভেতরে একটা পিস্টন রড থাকে, এই পিস্টন রডটা আপ অ্যান্ড ডাউন করে এবং সেই ক্যাভিটির মধ্যে একটা অয়েল ফ্লো কন্ডিশনিং থাকে। দ্যাট মিনস এর মধ্যে একটা ফ্লুইড বা লিকুইড আছে। আমাদের এই সিলোমিক যে ক্যাভিটি, হ্যাঁ, ইট ইজ অলসো ফ্লুইড ফিল্ড, দ্যাট মিনস সিলোমিক ফ্লুইড ইজ প্রেজেন্ট, অ্যান্ড হোয়েন এক্সটার্নাল শক অ্যারাইজ then it is uniformly distributed and the internal organs are not damaged. Okay, this is the main function of the coelomic part. This type of coelom is of various type. In the condition where the organism do not contain any coelomic cavity in the body. Okay, before this you have to you have to understand the basic criteria of coelom. First criteria, the organism must have to be triploblastic. Okay, and the coelomic cavity will be lined by coelomic epithelium, that means peritoneum, visceral and parietal peritoneum. Okay, this is the visceral peritoneum layer. Is that clear? If in any organism the visceral peritoneum is derived from mesodermal cells, then this type of coelom can be considered as a true coelom. Okay, in some organisms where there is no body cavity, okay, or fluid filled cavity, say as for in diploblast like Cnidaria and Ctenophora, there is no cavity. This is the ectoderm layer and this is the endoderm layer. In between this ectoderm and endoderm, a mesoglia is present, okay, which is actually filled by parenchymal cell, and this type of parenchymal cells are made up of connective tissue, mesenchymal cell and muscle fiber. This parenchymal cell restricts the movement of internal organs of this type of coelenterate and Cnidaria. So these are diploblastic acoelomate arrangement. Okay, and here in platyhelminth, that means in flatworm group, the ectodermal layer and endodermal layer. In between ectoderm and endoderm, another layer, mesoderm, is present, but the cavity is not present here. So this is the triploblastic acoelomate. You can see the cross section of the flatworm. Here, this is the outer body covering. This outer body covering forms the ectodermal part, and this is the inner digestive tract, forms the endodermal part. in between ectoderm and endoderm, you can see in red color the tissue filled region, that is mesoderm. There is no fluid filled body cavity. So this is the triploblastic acoelomate arrangement. In nematode, that means Ascalmenthes, here a body cavity is present. Okay, this is the cavity, but this cavity is not derived or formed from the mesodermal layer. Okay, this is the ectoderm portion, and here this is the endodermal layer. In between ectoderm and mesoderm, a cavity is present, but this type of cavity is actually derived from this blastocele in early embryonic development, and this blastocele is remain unchanged and present in adult organism. Okay, causes a hollow cavity, but cavity layer is formed by basal lamina cell. It is not mesodermal in origin, and this cavity is filled with a fluid. Actually, this type of body cavity is the modified blastocelic cavity. Okay, so this is not true coelom. It is a false body cavity, and this type of arrangement is known as pseudocoelom. So the nematode are the pseudocoelomate group. Okay, this body cavity is present in embryonic condition as a blastocele, or as well as in the adult organism, and in this cavity the organs remains present in free condition. Okay, this type of cavity is actually works as a hydrostatic skeleton to maintain the shape of the body and circulates all the nutrients of nematode group. Okay, here you can see the body wall musculature, and this is the intestine portion, that means gut. In between the ectoderm and endoderm, okay, this is the body cavity portion. Okay, this body cavity is not mesodermal in origin. So the nematode roundworm are the pseudocoelomate group, and in pseudocoelom the mesodermal cell are present as mesodermal pouch. You can see these are the mesodermal pouch, and this type of cavity is the pseudocoelomic space where the internal organs of that organisms remain suspended in free condition. And the organisms where the body cavity is actually derived from mesodermal cell by forming the visceral and parietal peritoneum. That means it is a perivisceral cavity. This is the ectoderm part. Okay, this is the ectoderm part, and this is the endoderm part, and this is the gut. These are the mesodermal cell. Clear? And in between ectoderm and endoderm, you can see that a body cavity is present. This is the body cavity, and this cavity is known as coelomic part. That means coelom is continuous to the gut. Okay, but in pseudocoelomate condition, the body cavity is continuous to the gut, but here the endoderm, then mesodermal cell, then the body cavity, then mesoderm and ectoderm. This type of arrangement is present, and this cavity is filled with a specific type of fluid, that is coelomic fluid. Okay. Here the mesodermal cell, from this mesodermal cell, both these outer part, that means parietal peritoneum, okay, is produced, and this parietal peritoneum is produced from somatic mesodermal cell, okay, and also from this mesoderm the visceral peritoneum cells are produced, and visceral peritoneums are derived from the splanchic mesodermal cell. This is the visceral peritoneum, and this cavity is the perivisceral cavity. Okay, and this gut is continuous with a specific type of cell. This is known as mesentery. Okay, mesentery helps to connect the gut and other internal organs, okay, to the body. Okay, actually, in case of true coelomate or eucoelomate condition, this is the true coelomate arrangement. This is the outer body wall musculature, it is the intestinal portion, and the cavity, this cavity, okay, this is the coelomic cavity arrangement. Okay, this true coelomate arrangement, all the internal organs remain suspended freely. Okay, this type of coelomic cavity are filled with coelomic fluid. This coelomic fluid are actually produced from this type of peritoneum epithelium, okay, and which acts as a shock absorber. So all the organs in that organisms are not damaged. Is it clear? And the organism, that means the annelid to caudate. Annelid is the first eucoelomate, that means true coelom is developed. Okay, annelid to caudate, that means up to human, all the organisms are true coelomate. In arthropod and mollusks, the coelomic cavity is filled with blood. This is known as hemocoelic arrangement or hemocoelom. Coelom development is of two types. Okay, this is the blastula stage, and here this is the blastopore. Blastopore is formed just opposite to the site of sperm entry. Okay, this is the ectoderm portion. Here the cells during blastulation are enfolded and form the endodermal portion, and this is the embryonic gut, that means archenteron part. Okay, and the organism, a group of organisms, okay, where this blastopore at first converted to mouth. Okay, this is the blastopore, and this blastopore forms the mouth part first, and anus secondarily developed. Is that clear? As mouth developed first, so this group of organisms are known as protostomes. Okay, in protostomia condition, the early mesodermal cell, this split into mesodermal wall. Let's try to understand. These are gathered and forms a pouch-like structure, and this archenteron forms the endodermal part, and this is the ectodermal part. Okay, and the blastocele is gradually replaced by these type of mesodermal cells. Okay, and these are actually the coelomic epithelial layer and forms the coelom. Okay, this type of pouch is actually known as schizocilous pouch, and this type group of organism are known as schizocoelomate generally. Okay, Annelida, Arthropoda and Mollusca. Okay, this group of organisms are schizocoelomate group. Okay, and in different condition in schizocoelomate group, okay, the cleavage is done by spiral cleavage method. In some groups radial cleavage occurs, and here the blastopore actually forms the anus first. Okay, from blastopore the anus is developed first and mouth develops last secondarily. As mouth developed last, so these groups of organisms are known as deuterostome. In this group of organisms, this is the gut wall, that means archenteron. The early mesodermal cells, these will attach to the enteron or gut portion. Later on, these will pinch off and separated from this gut portion. Okay, these are the separated pouch. As this enteron, that means early mesodermal cell in early embryonic development, these are attached to the gut portion. During the embryonic developmental procedure, these will separate, okay, and form the pouch like coelomic structure, and from where the coelom is developed. Okay, as it is splitted from the primitive gut or archenteron, this type of coelom is known as enterocoelomate. Okay, and this type of arrangement are known as, okay, enterocoelous type of arrangement. And here the remaining groups of organism, that means echinodermata, hemichordata, urochordata, cephalochordata, as well as the total vertebrate groups are the enterocoelous group. So let's try to understand the different pattern of coelom development. Okay, in sponge there is no tissue formation. Okay, from sponge to this flatworm, these are acoelomate group. Okay, porifera, nidaria, ctenophora and platyhelminths, these are the acoelomate group. And the nematode, that means roundworm, are the pseudocoelomate group. Okay, and from annelida to caudate, these are the eucoelomate, that means true coelom group. Flatworm, nematode, annelida, arthropod and mollusca, these are the protostome group. That means blastopores is converted to mouth first, then anus is secondarily developed. And in echinoderm at caudate group, blastopore is converted to anus first, then mouth developed last. So these are the deuterostome. And the annelid, arthropod and mollusks, these are the schizocoelous group. Okay, echinodermata, hemichordata, urochordata, cephalochordata, and vertebrata. That means echinoderm and chordate group are the enterocoelous group.